Hello, and today we're going to talk about synapses and neurotransmitters. So we want to talk about what a synapse is, and you should already know what a synapse is because we did this with the muscle, with a neuromuscular junction. That was a synapse, and that's just simply the junction where you transfer information from another neuron to an effector, and that effector can be another neuron or a muscle or a gland. So you have your presynaptic neuron, which is bringing information or impulses to the synapse, and then the postsynaptic side, which is either a neuron or a muscle or a gland, which takes the information away from the synapse. So if it's a neuron, it's going to be another action potential. So you would have, in this picture, you see an axon coming in and synapsing on the cell body of this neuron, and then this neuron will then fire and send information away down its axon. So again, a synapse is simply the junction between a neuron and either another neuron or a muscle or a gland. And I just put this picture in here so that you could see this is an electron micrograph picture, basically a microscope at high resolution, and you can actually see the little synaptic knobs synapsing on the cell body of this neuron. I just thought it was a neat picture. So when we think about synapses, we've covered chemical synapses, which I'm going to talk about again, but there are things called electrical synapses. These are not as common as chemical synapses, but if you remember, the neuromuscular junction was a chemical synapse where you had your axon terminal and then you had your muscle on the other side, and there was literally a space or a cleft between the two sides. In electrical synapse, these neurons are joined together. They're physically in contact so that the electrical signal can simply spread from one neuron to the next. And these are really important because it's really fast. You don't have to wait for the neurotransmitter to be released and to diffuse across the synaptic cleft and bind to a receptor and open a channel and then cause something to happen. This is just really quick. And you're going to see it in embryonic nervous tissue, which makes a lot of sense because this is early development and you don't really have all your stuff yet to make a synapse. It's very complicated. You don't have everything in place yet. And then parts of your brain use these electrical synapses. But most of our synapses are chemical synapses. And the chemical synapse is what we're used to. It's the real common thing we talked about, like a neuromuscular junction. And these are synapses that use a chemical as the signal, where you're going to release a neurotransmitter and then have a receptor for that neurotransmitter. So just like in the neuromuscular junction, your neuron released acetylcholine, and then the muscle had the little acetylcholine receptor allowing for some kind of effect to occur. Well, that's a chemical synapse. So you have your axon terminal of your presynaptic neuron, has a vesicle containing a neurotransmitter, and there's lots of neurotransmitters. And on the other side, on the postsynaptic side, you have a receptor that binds that neurotransmitter, usually maybe a channel or a protein, and when it's bound, something happens. So this is just a review. Same thing we did with a neuromuscular junction. This is just an axon synapsing with another neuron. So this would be a neuron to neuron synapse. The same things apply as what we learned about in the neuromuscular junction. Your action potential or impulse travels down the axon. That causes a voltage-gated calcium channel to open. That causes your vesicles containing a neurotransmitter to exocytose their contents out into the synaptic cleft. They diffuse across the cleft, bind to a receptor on the membrane, and then cause some kind of effect. And then those neurotransmitters either diffuse away or are broken down with an enzyme. So this is a review. You already know this from your neuromuscular junction. I will test you on it again because I don't want you to forget about it. And one of the reasons I don't want you to forget is we're going to talk about the endocrine system in our next semester. And so we're going to talk about the hormones and binding receptors, kind of like neurotransmitters bind receptors. So we want to kind of keep these thought processes going as we move forward into our physiology. We're kind of building on what we've learned. So when a neurotransmitter binds to a receptor, it can do one of two things. It can excite, it can inhibit, or it can do both. So if a neurotransmitter is excitatory, that means when it binds, it opens up a sodium channel, allowing for depolarization, which will be excitatory. You would get an action potential, you would get an impulse, you would get a signal. If it's inhibitory, it may open a chloride channel, or it may affect the potassium flow. And if you open the chloride channels or allow potassium to move, you're going to have hyperpolarization, which, if you remember your action potential, this will be the hyperpolarization. If you're going away from threshold, so if you're taking the membrane potential even further down, more negative, then it's going to be harder for that action potential to occur. So it's going to hyperpolarize the membrane. So it can either excite it by depolarizing slightly the membrane or inhibit it. Some neurotransmitters do both. It just depends on the type of receptor. So if, let's just say neurotransmitter, let's just give it the word neurotransmitter. If it was to bind a receptor, say, in your skin, I'm just making stuff up, okay? It might be excitatory. If it bound a receptor, say, in your big toe, it might be inhibitory, depending on the type of receptor. Was it a sodium channel that it bound, or was it a potassium or chloride channel that it bound? And when a neurotransmitter binds, the strength of the signal is determined by how much you release, how much neurotransmitter is released into the synapse, and how long it's there. So the longer the neurotransmitter's in the synapse, the more chance it has to bind and stay bound and cause either an excitatory or an inhibitory effect. You don't want your neurotransmitters to stay in there too long, and so typically within a couple of milliseconds you get rid of it. And you get rid of it by breaking it down with an enzyme. If you remember acetylcholine, we use acetylcholinesterase. So you have some kind of enzyme that breaks it down. You actually have some astrocytes that will take it back up. Sometimes the neuron will have little pumps that basically channels that pump it back up into the axon terminal, or it may just diffuse away. So the point, you want to get rid of the neurotransmitter so that the effect doesn't last forever. Now, some drugs, so pharmacologic drugs, recreational drugs, things like that, what they do is they act by allowing the neurotransmitter to stay in the synapse longer. They either stop the enzyme from working, or they stop the reuptake so that the neurotransmitter stays in the area longer. And so, for example, maybe something maybe allowing serotonin, which is a neurotransmitter that makes you feel good. If it stays in the synapse longer, you'll feel better. So some drugs work by blocking or changing how long the neurotransmitter can stay in the synapse. So when we talk about neurotransmitters, we can classify them chemically, what they look like structurally, what they're made, their chemical composition is. So we have one, acetylcholine, which we've talked about. It's actually made by an enzyme, choline acetyltransferase, which basically makes the neurotransmitter. And it's made in the neurons that bind or synapse with the neuromuscular junction, and also some autonomic nervous system neurons. So if you think back to that first diagram, central nervous system, peripheral nervous system, and I told you guys to be able to fill out the little cascade, your autonomic nervous system will release acetylcholine. Some of the neurons do. You use acetylcholinesterase to break it down, and its main action is voluntary muscle movement. But again, we'll talk about some other things it does later on. You have some biogenic amines, your catecholamines. Examples of those are dopamine, norepinephrine, and epinephrine. You may have heard of these named adrenaline and noradrenaline. Okay, so dopamine, norepinephrine, and epinephrine. Just some brief things that they do. Dopamine allows you to feel emotionally aroused, feel good. Norepinephrine we use in wakefulness. Epinephrine increases heart rate and blood pressure. So if you've ever heard of the word adrenaline, you get an adrenaline rush, your heart rate goes up. So yeah, epinephrine is a neurotransmitter that does that. If you think back to your cascade again, I said we were going to talk about your sympathetic and parasympathetic nervous systems, your fight and flight. Well, epinephrine is one of the neurotransmitters that plays a role in that fight or flight, and we'll talk about that a little later on as well. You have some things called indolamines, serotonin and histamine. Serotonin plays a role in memory and wakefulness and temperature regulation. And then histamines, and they play a role in wakefulness and inflammation. So if you take something like Benadryl, which is an antihistamine, a lot of times it makes you sleepy, okay, because you're blocking the histamine's action. So those are some chemical classes. Some other chemicals are your amino acids, and for example, GABA and glycine. Now, GABA stands for gamma aminobutyric acid, but you can say GABA. And what it does is inhibits motor neurons, so it kind of slows them down so they don't get a signal to contract. So it slows down the muscle, the motor neurons to muscles and things like that. And so it's enhanced by alcohol. So if you drink a lot of alcohol, it causes you to have the GABA to work better, which would slow down your motor neurons, which leads you to think, okay, that's why you're kind of slow. Your reaction time's a little slow when you're drinking. Glycine plays a role in reflexes. We'll talk a little bit about that later. You have some glutamate or neuropeptides. One of those is substance P. It's part of your pain signaling neurotransmitters. Endorphins, which are your natural opiates. So like opium, this is very similar in structure to the opiates. And endorphins make you feel better. And they do that by reducing your pain perception. So the runner's high, yeah, your body still feels pretty bad. You're still in pain, but the endorphins allow you not to perceive it as much. So it gives you that high feeling. You have some purines like ATP. These work in the central and peripheral nervous system. And again, they're for pain. And then you have some gases, for example, nitric oxide. And nitric oxide works as a vasodilator. It's actually a gas. It causes the arteries and capillaries to open up. And so if you open up the capillaries, that allows more blood to go into an area. And so, like for example, in the erection of the penis, nitric oxide helps in the engorgement of the penis by vasodilating the vessels. And so things like Viagra work by increasing the amount of nitric oxide in the area and increasing vasodilation. So we use it also in memory and learning. We can classify neurotransmitters functionally. Again, they can be excitatory or they can be inhibitory. And this really depends on what the receptor is. So if the receptor is a sodium channel or a potassium channel or a chloride channel. All right. GABA and glycine are almost always inhibitory, so they're going to open up a channel that causes your membrane to hyperpolarize. And glutamate is typically excitatory. It opens up a channel that allows your membrane to depolarize, so it becomes more excitable. And then acetylcholine, it does both, but it just depends on what the receptor. So at the neuromuscular junction, it's excitatory, but in the cardiac muscle, it's inhibitory. So it depends on the receptor. So the neurotransmitter itself is exactly the same. It's the receptor that's different. Let's talk about how neurotransmitters work. They can work directly, what we call the direct action. And this is just what you've already learned. Basically, a neurotransmitter binds to a channel, and that receptor channel opens and allows ions to flow. Okay, and this is really fast. So, example, acetylcholine binds to the neuromuscular junction receptor, opens up the sodium-potassium channel, depolarizes the membrane, and you can start having an actual potential move down your sarcolemma. So that's fast. Acetylcholine and amino acids both work like that. So just, again, this is review. So here's your channel. Ions can't flow, they're blocked. If your ligand or neurotransmitter binds that receptor, it opens up the channel and now ions can flow. That's your direct action. Again, some of this is going to be review today, some of it's new. But you can also have an indirect, and this is where the neurotransmitter binds to a receptor in the plasma membrane. Basically, it's a protein, and that causes a cascade. And this cascade causes the production of something called a second messenger, and the second messenger is what actually causes the response. Okay, so you're going to have, I'm going to show you a picture of this. But second messengers can open and close channels, they can activate enzymes, they can change protein structure, they can activate genes, and so these are slower. These take a while. And some examples of these are your amines, your neuropeptides, and your gases. So what does this look like? So your neurotransmitter is going to bind a protein, which is your G protein receptor in your plasma membrane. And I don't necessarily want you to memorize all these steps, but I do want you to know that when you bind this receptor, this G protein is then activated. And you get this activation of GTP, and which is kind of like ATP, just has a G in it. And so this GTP will then bind adenylate cyclase, which is an enzyme. ASE is an enzyme. Well, this is key. This adenylate cyclase then converts ATP to cyclic AMP. This is your second messenger, okay? So this is what you want. You want your cyclic AMP to be formed. And the cyclic AMP, depending on the cell, depending on the neurotransmitter, depending on what the effect's going to be, can do several things. It can either open and close a channel, activate another enzyme, or it can work at the nuclear level and activate genes and cause protein synthesis. So you can see it takes a little bit longer for this to happen than in the direct action. So the take-home message is you need to bind a neurotransmitter to a G protein receptor, activate the G protein, activate adenylate cyclase, convert ATP to cyclic AMP, and then cyclic AMP can then do any one of these different effects, okay? And that's the second messenger system, a little bit slower. All right, so how do drugs and neurotransmitters work together? So a lot of the drugs that we take, especially our mood modulation drugs, antidepressants, things like that, work by changing how the neurotransmitters either bind or how long they stay in the synapse or how they're degraded or not degraded. Okay, so we're usually looking at somehow changing the way the neurotransmitter is able to function. So one class are your SSRIs, which are your selective serotonin reuptake inhibitors. And so reuptake inhibitor. So remember your axon. Okay, so let's say serotonin's out in the membrane and it's able to bind its receptor. Well, if you have a reuptake inhibitor, that means it's not allowed to be taken back up into the axon terminal. It's going to stay in the synapse longer, and it allows you to feel happy. So this is an example: Lexapro, Prozac, Paxil, Zoloft. They all work like that. They just allow the neurotransmitter to stay in the synapse longer. MAOIs, you've probably heard of this. If you're taking an MAOI, don't take, drug X, Y, or Z. This is a monoamine oxidase inhibitor. And monoamine oxidase is an enzyme that breaks down neurotransmitters, like acetylcholinesterase. So these are some examples of monoamine oxidase inhibitors, or MAOIs. And so if you stop this enzyme, if you inhibit monoamine oxidase, then the neurotransmitter is not broken down. It stays in the synapse. You get more stimulation, and you feel happy, okay? Another way, amphetamines, these are some of your psychostimulants like Adderall, and they can modulate the effects, either let the serotonin bind more robustly to the receptor, norepinephrine, dopamine. Basically, it modulates how these neurotransmitters bind to the receptor. And because it can work on so many different neurotransmitters, that's why it can be hard to judge exactly how and who will be helped with Adderall. And the same thing with these selective serotonin reuptake inhibitors and MAOIs. Everybody's wired a little bit differently chemically, so sometimes you'll need to try 1 or 2 or 3 drugs before you find the one that works the best for you or your patient. Another thing, a recreational drug, I want to talk about cocaine. And cocaine, the way it works is it blocks dopamine reuptake. So just like the SSRIs, cocaine causes dopamine to stay in the synapse longer, and because it stays in the synapse longer, it makes you feel happy. So the cocaine's not actually binding your receptors. It's not actually kind of triggering a good response. It's allowing your own dopamine to do that for you, okay? So there's something called the mouse party, and there's a link to it in your blackboard. And I want you to go through the mouse party, and you'll play with the little mice and see what the drugs do, how the drugs work. So as you watch the video, take notes, pay attention to the name of the drug, which neurotransmitter it's affecting, and how does it affect it? Does it block its reuptake? Does it block the receptor? And what is the overall effect? It's a fun game, take you about 20 minutes, but be sure to take some notes because you will see some questions on the exam about these recreational drugs. There's LSD, ecstasy, alcohol, marijuana. So again, take some time to play with that and learn about how the recreational drugs work in changing the chemical neurotransmitters in your brain. All right, that's all I have for you today. Thank you for your attention, and I'll see you again later.